Crohn's disease exclusion diet plus partial enteral nutrition induces sustained remission in a randomized controlled trial by Levine and colleagues 2019. This video digest presents the study rationale, the design and methodologies, the main results, and the significance of the findings. Crohn's disease, one of the clinical manifestations of inflammatory bowel diseases, is associated with an alteration of the gut microbiome, as well as an impairment of the intestinal barrier integrity. Exclusive enteral nutrition is recommended as first-line treatment for children with mild to moderate Crohn's disease, based on its proven superiority with respect to oral corticosteroids in inducing clinical remission and mucosal healing with no major side effects. Nevertheless, the need of nasogastric tube feeding and the prohibition from eating any other solid food for 6 to 8 weeks limits exclusive enteral nutrition clinical application in a wider population. Crohn's disease exclusion diet is a whole food diet which has been designed to eliminate specific components that could trigger inflammation, microbiome dysbiosis, and affect the integrity of the intestinal barrier. The study by Levine and colleagues aims at assessing the tolerance and the efficacy in inducing and sustaining corticosteroid-free remission through Crohn's disease exclusion diet coupled with partial enteral nutrition, compared with gold standard exclusive enteral nutrition in pediatric patients affected by Crohn's disease. Patients in several pediatric clinics in Israel and Canada were enrolled in this multi-center, randomized, controlled clinical trial. If they were aged between 4 and 18 years, diagnosed with mild to moderate luminal Crohn's disease, and presenting active inflammation at enrollment. Recent use of steroids, initiation or dose adjustment for immunomodulators, past or current biologics use, primary colonic disease, and active perianal disease were all reasons for exclusion. Patients were randomized in 2 groups. Group 1 receiving CDED and 50% PEN for the first 6 weeks, then CDED and 25% PEN for the next 6 weeks, and Group 2 receiving EEN for 6 weeks, followed by 25% PEN during weeks 6 to 12, with gradual reintroduction of foods between weeks 6 and 9. Patients were visited at baseline and at weeks 3, 6, and 12. Pediatric Activity Index was measured at each visit. Laboratory tests included complete blood count, erythrocyte sedimentation rate, C-reactive protein, albumin, and fecal calprotectin. Lactulose mannitol test was used to assess intestinal permeability at weeks 0 and 3. Microbiome analysis was performed by sequencing 16S ribosomal RNA gene on stool samples at weeks 0, 6, and 12. Since dysbiosis at a phylum level in CD is characterized by increased proteobacteria and decreased firmicutes, CDED includes 5 mandatory foods to provide protein, nutritional balance, and enhance firmicutes, while improving intestinal barrier function. The second phase step-down diet was characterized by a high exposure to fruits, vegetables, and legumes. The primary study endpoint was patients' tolerance to the diet by week 6, defined by the withdrawal from the study because of patients' refusal to continue the diet. Secondary endpoints were assessed at 6 and 12 weeks and included response to the diet and clinical remission, assessment of inflammatory markers, and adherence to treatment. 78 patients were randomized to the CDED plus PEN and EEN groups. The sample size calculation was performed based on pilot data and published results, assuming an 80% power and type 1 error probability of 0.05. No significant differences were observed in baseline patient demographics. The primary endpoint was significantly different between the 2 groups, favoring CDED plus PEN over EEN. No significant differences were observed at week 6 for response and corticosteroid-free remission, even considering the more stringent PCDAI is less than 10. Good compliance was observed for both groups after 48 hours of treatment. PCDAI and inflammation markers decreased significantly in both groups after 6 weeks of diet. No significant differences were observed at week 6 for response and corticosteroid-free remission, even considering the more stringent PCDAI is less than 10. Good compliance was observed for both groups after 48 hours of treatment. CDED plus PEN patients showed a higher sustained remission at 12 weeks than EEN patients. This result was confirmed also in a subgroup of CDED plus PEN patients who achieved already corticosteroid-free remission by week 6, and for which the likelihood of maintaining remission at 12 weeks was higher than for EEN patients. Fecal calprotectin was very high in both groups at baseline. A significant decrease was observed for both groups at 6 weeks, but while this decrease continued till 12 weeks for the CDED plus PEN regimen, in the EEN group a non-significant increase was observed at 12 weeks. CDED plus PEN regimen increased the proportion of patients with normal intestinal permeability at 3 weeks, while for EEN patients this number remained unchanged. Microbiome analysis on stool samples from patients achieving remission at week 6 revealed that the pattern of taxa between the two groups differed significantly. CDED plus PEN microbiome composition continued to change between week 6 and 12, while in the EEN group the pattern at week 12 was similar to pre-treatment levels. Protobacteria decreased with both regimens, except for the EEN group at week 12. Patients who did not achieve remission at week 6, non-responders, showed less changes in bacterial composition than those who achieved remission at week 6. The main microbiome features of non-responders at 6 weeks, irrespective of diet, were an overall lower change in bacterial composition and more gamma proteobacteria. In summary, the use of CDED plus PEN compared to EEN in pediatric CD patients showed a better tolerance to the diet, a better sustained clinical response at 12 weeks, an improved intestinal permeability, and a beneficial effect on microbiome composition. Furthermore, the use of CDED plus PEN was associated with corticosteroid-free remission at 6 weeks in 75% of patients, and a high compliance rate despite the food restriction. Overall, the study by Levine and colleagues supports the use of CDED plus PEN as first-line treatment for children with luminal mild to moderate Crohn's disease, and opens new perspectives for the use of diet in combination with drugs to optimize CD therapy.